Okay, folks. Hi, how's it going? I can see that a few people have got some questions there. Never mind. Here I'm now. Okay, yes, I'm here. Okay, so regarding specific questions on the exam, it's best to email me about those, okay? Sorry, just looking at the chat here. Okay, I'm going to crack straight on because we have quite a lot to go through. Okay. First thing I've got up here is just overall results from midterm 1. So you should be able to see in the grade center and Blackboard how it broke down. It was pretty similar to midterm. Midterm 2 was pretty similar to midterm 1. The mean was 71% and the median was 71%. So it was slightly higher, actually, a few percent higher than. So that just gives you the general breakdown. Okay. Hello. Okay, people saying hello. Anyway, so that's an overview for you. What else have I got for you here? A few people have been asking about, what's the breakdown of my questions? This is just a reminder of what was in the syllabus, okay? So midterm 1 is 15% of your overall score for the whole course. Midterm 2 is 15%, that's what we've just done. And the final is 15% as well. Okay, if you think you've got enough marks to pass the course and you're not interested in doing better, so you think you don't need to turn up to the final exam, you still need to turn up to it to be able to get a grade, okay? Otherwise you get an incomplete and have to do it within the next semester. Quizzes and iClicker is going to be 15% in total, okay? I know that iClicker is not syncing with Achieve at the moment. I do have your scores, the scores that you have in your app. I see those as well, and I download them and back them up regularly. It's not syncing because my iClicker account is through a different email address, registered through a different email address than my Achieve. Achieve doesn't seem to like that, and it won't allow me to sync at the moment. Okay, so most of those marks will be based on quizzes because that's where most of the questions are. iClickers come into play as well. There'll be some dropping of, if you didn't turn up to, I think Dr. Spagnola had up to 3 iClicker sessions would drop in, he has in there.
Professor?
Yes, sorry.
So for iClicker, is it based on whether you pass or is it based on your percentage of what you got right or wrong?
Percentage. Well, the way that I've set mine up is that overall it's based on the iClicker percentage. It's not just a yes or no. And I give you the way I'll set it up in the most recent lectures and from now on is that if you just turn up and log in, and we're not doing it today, just in case you're wondering about that. Hopefully that doesn't mean 100 people will disappear. But you get 1 point for just like arriving, right, and logging in. And then for answering questions, I think I've set it up. I mentioned it last time, like you get 0.8% for just trying, 0.8 of a point for just trying, and then 0.2 or maybe it's 0.1, but a small fraction on top of that if you get it correct. So most of it, how much of the iClicker is worth, the whole 15%, it'll depend at the end of semester of how many questions spread across everything, but the iClicker is going to be a small percentage because we have so many more questions within the quizzes, okay? I don't know exactly, I'm guessing, but don't hold me to it. It'll be around the equivalent of like one of the quizzes or something, okay? I'm guessing. So just a reminder of the other part, the 40% is coming from your lab. So, I don't have access, well, I do have access, but I haven't looked at your lab grade, so that's something you can discuss with a lab instructor. Please, can you remove the exam grade from Achieve? so we can see our real achieved grade. That's a Dr. Spagnolo issue because he set that up. I don't want to interfere with his exams. But you can look at your individual scores, right? For individual columns, for individual performances. Does this turn, okay, I'll come to the thing. Overall final letter grade is an adjustment for balance. That's the curving question you just asked. We don't do that until the end of the semester because we need to put all the different marks scores together to see if there's anything that's like really out of whack, you know, like one set lab section, for example, got like a really low score to compare to everybody else. So that's the kind of thing that we do to adjust for those imbalances to make sure that people don't get unfairly you know, hit just because of some particular reason. Your mark will only, if a curving does occur within a section, your mark will only ever go up or to stay the same at the worst scenario. It'll never go down, okay? So those, you know, don't ask specifically now, is this curved, because we don't know until we put the mix together at the end, okay? They curved down in 105 last semester. Okay, well, we don't intend doing that, okay? Is there usually a curve? I don't know. Don't ask me about curving. I won't know until the end of the semester, okay? Okay, also, I just want to point out I've updated the syllabus because, you know, well, for a couple of reasons. One of them is that when the syllabus went out at the beginning of the semester, the date of the final had not been put in the college schedule yet. That's something we don't decide. That's something the college decides. So take note, that is May 20th at 11 a.m. to 1 p.m. is the slot they've given. That is something I can't change. Okay, that's a fixed date. So if you work and your boss says so-and-so, please, you know, try and make accommodations for that, okay, etc. That's the date of the final. I can't control that. Also note that chapter 2 is gone because we're a little bit behind, which is fine. I'm not worried about that whatsoever at all, because that's a chapter that's covered in Bio 105. Is the final cumulative? No. It says that in the syllabus, okay? So note here, final exam, covering lectures 8 to 26, okay? So if, you know, if there's something covered in those lectures that was covered in a previous lecture and it's covered within 18 to 26, well, it's still in 18 to 26, you know, if some concept happens to be repeated twice. But it's not like you're testing on the whole syllabus all over again, okay? Like I said, chapter 2 is gone. That's developmental biology. You do that in 105 anyway, so there's no real reason for doing it all over again, okay? And it's a bit of a pain of a chapter, to tell you the truth. So will it be part 4 test again? Yes, I suspect so. I will see how it goes. Everyone have different opinions, but I think most people think that's way better. Okay, so moving on next. Yes, I'm muted, sorry. Most people found the 4-section format preferable. Not everybody, but it's 250 people, so I'm afraid I can't satisfy everybody, right? We've got to go with the way that things work most efficiently. Okay, the reason, so people have asked about looking back on questions. That was a setting I made, okay, a choice. That can be done in Achieve or Blackboard. The reason for that I didn't allow backtracking to questions is for reasons I outlined in the lectures. Some of those reasons are fairly obvious, right, when we're doing a remote exam. So this lecture, we're moving on to look at the deuterostomes. Okay. So let's just take a little reminder where we are in the grand scheme of things. We've been talking about animals since I took over the lectures effectively. So remember, there was a whole load of bits before this of all organisms when they started evolving, but now we're looking at animals, and we've covered these organisms. We've just finished talking about the protostomes. Okay, we had a lecture on Lophotrochozoans. We had a lecture on the ecdysozoans, the most diverse group of organisms on Earth, with the arthropods in there. And now we're moving on to the deuterostomes. Okay, so these are all part of this. Let me just get my pointer going. If you remember, the protostomes and the deuterostomes are part of this group down here that are triple blastic animals. That means they have 3 tissue layers, okay? An endoderm, ectoderm, and mesoderm. And they have bilateral symmetry. So both the protostomes and the deuterostomes, as a synapomorphy of that clade, have bilateral symmetry. So one of the main features that distinguishes these deuterostomes is this aspect here of the blastopore in the embryo developing into an anus rather than the mouth. Okay, deuterostome means mouth second. Okay, it's not 100% distinction between the groups, as we often find with these things, but that is one of the key features they put in here. So what do these organisms include? 3 major clades, 3 major groups of animals that over the next couple of lectures we'll talk about. One of them is the echinoderms. That includes things like starfish, sea cucumbers. These are things called crinoids, sea feathers, and sea urchins, and brittle stars, okay? We'll be talking about those in this lecture. It also includes a small group of marine organisms called the hemichordates, okay? We'll discuss those in this lecture as well. We'll touch upon the chordates at this lecture and then do them, cover those next lecture as well. The chordates includes things like us, right? Vertebrates, but it also includes, this is a hagfish, this is, well, etc. So we'll come across all of this, and we'll talk about these in the next couple of lectures as well. So what features, just checking the chat here, okay. What features are important or distinguish the deuterostomes? A lot of the features are based upon early embryonic features. One of them is that they have radial cleavage in the early stages of the development of their embryo. Okay, so when you have a fertilized egg, it starts dividing into multiple cells, and it creates this pattern here that's shown on the bottom in general in the ancestral form, where these cells are stacked one on top of each other, okay, in this circular form. Whereas when we talked about the protostomes, the sort of general feature that they have is this spiral cleavage, where these cells are at a sort of oblique angle as they divide off. The second key feature is that this blastopore that invaginates here within this hollow ball of cells, that will eventually develop into the anus, and the mouth develops at the second end, okay, the deuterostome second, mouth second. That is the opposite from the general pattern that occurs within the protostomes, okay. The third key feature is that the coelom develops from mesodermal pockets. Well, what does that mean? The C side and mesodermal tissue. And this diagram shows how that occurs. The mesoderm are these red blobs here of cells. You froze, Professor. Okay. I will refresh, or am I back up again? I thought it was just, okay. Am I still frozen? You're good now? No. Okay, good. Right, I'll try not to freeze again. Okay. Okay, so coelom, yes, I was talking about coelom. So that develops from mesodermal tissue, okay? And that's these red blobs here. No, the class is up, I'm not doing iClicker this session, Lydia. These mesodermal tissues in the deuterostomes, they develop from this internal pocket here, okay, called the alcantaron. And then the coelom develops as a pocket within inside that ball of cells, okay, or it pinches off. It's like pinching off of that alcantaron. Compare that with the protostomes, where you have this ball of cells here, and then the coelom sort of expands from within, okay? So a different pattern in the way that the coelom develops. Radial cleavage in most deuterostomes is indeterminate, okay? What does that mean? They have indeterminate development. What that means is that when you get to an early embryonic stage, so this 4-cell stage, if you took each one of those cells and separated it, and then let them carry on developing, they could still develop into an entire new organism, okay? That is different from in the protostomes that have determinate cleavage, where if you did this at this point, that wouldn't occur. You wouldn't get the full organism developing from them. Embryo development patterns are not the strongest evidence for common ancestry in the deuterostomes, though. Okay, we're kind of getting used to this theme that there are these many features that are generally true for a group, but often there are exceptions because there have been modifications on the way, you know, after being derived, for example. And the strongest evidence comes from genomic sequencing, true in all of the clades that we've been looking at. So an example with some of the features I just talked about, radial cleavage occurs in some other groups. Okay, I think we talked about the arrow worms, the ketonasths that have radial cleavage, which are a protostome. Development of blastopore into an anus may be ancestral of all bilaterian animals. So both the protostomes and the deuterostomes, it might actually be the ancestral condition there. So it's thought. The earliest fossil records of deuterostomes comes from about 520 million years ago from a very well studied fossil area in China. And some of the features that they have is that these fossils are bilaterally symmetrical, not surprising, because that's a synapomorphy of both the protostomes and the deuterostomes. They also have a pharynx with gill slits, and we'll be talking about those quite a bit in the next lecture, well, particularly next lecture. So that the pharynx is that sort of area, your throat area, effectively. And gill slits, imagine like on a fish, right? They've got these little openings so that you can take in water through your mouth and it can pass back out again. You can pump it and it passes back out through those gill slits. They're also segmented, these fossils, but that is not a distinction of the deuterostomes in general, because remember we talked about quite a few of the protostomes that have segmentation, such as the annelid worms, the arthropods, okay? But not every day, modern day deuterostome has all of these features. Some of these things are lost, like we don't have gill slits that are evident to us, right? These pharyngeal gill slits, except for embryonically. They do actually occur for a short period embryonically in humans. So this is the current view of the deuterostome phylogeny, okay, which groups evolved from common ancestors. I'm not going to go through it now, but there's actually quite a good video that I put here that's part of the, the book. It's a 6-minute video that is a good kind of overview and introduction to it. No iClicker today, okay? In answer to that multiple asked question, okay? I advise, it's good to look at that video in your own time, okay? So here we're talking about a common ancestor that had bilateral symmetry and pharyngeal gill slits, okay? And two major groups, one at the top here that includes the echinoderms, starfish and things like that, and this small group, the hemichordates. And they are classed together in this, what's called the ambulacrarians, okay? Their common feature is that they have a ciliated larva. Then we have the chordates, which is the main topic of next lecture. That involves ultimately vertebrates, including us. So let's look at these ambulacrarians, okay? These are deuterostomes with bilateral, a bilaterally symmetrical larva. Okay, so they have a larva. Remember, that's the larval stage is something that looks different from the adult stage that the egg develops into. Often it's very mobile, so it can move around in seawater, for example. It's planktonic, and then it undergoes a metamorphosis into that adult form. So this is a ciliated larva that they have. With two major groups, the echinoderms and the hemichordates. This group in general is restricted to marine habitats. And there's also, the book mentions a couple of other sort of really poorly studied groups down here, the xenotubelids and the aseals that may be a sister group of the ambulacrarians. So, who come up here somewhere, but it's very uncertain at the moment. Or they may be a sister group of the bilaterians in general, so a sister group of the deuterostomes and the protostomes. So uncertain. But the main two that we're interested in is the echinoderms and the hemichordates. So we'll talk about the hemichordates first because they're a smaller group, and get those out of the way. These are worm-like marine deuterostomes, and there are two major groups called the acorn worms, only about 90 described living species, and the pterobranchs, okay, only about 30 or so described living species. So this down here is a picture of an acorn worm on the bottom left. They have bilateral symmetry, as is typical of most deuterostomes. And their body is divided into 3 different regions. The proboscis at the front, or the anterior end. Now, some people say proboscis, and I think they actually say that in the video I mentioned earlier on. I say proboscis, it's depends maybe where you come from or how you do it. The collar region, which is this little region right here. Now this is the collar here, and that's where the mouth is. It's at the anterior end of that collar region. Okay, this here is the proboscis, and then there's the trunk region that basically contains the rest of the organism. Okay, back here. These things can be up to 2 meters long, so quite long. They burrow in soft marine sediments, and their digestive tract includes the mouth, the pharynx, so that's the throat area basically. And you see they've got these gill slits that I mentioned in the pharynx, and then the intestine, okay? So the mouth is kind of in between the collar and the proboscis. Yes, I think in the book it actually says it's part of the collar officially. But it's in this diagram, at least it's at the anterior end, okay? So the pharynx opens to the outside via these pharyngeal gill slits. So in other words, water can be taken in through the mouth and then passed out through these, what they're calling gill pores here, okay? And there's vascularized tissue around the slits. That means that it means it has a good blood supply, so it's good for gas exchange. That water flow is being used for gas exchange, and they consume food with a large ciliated proboscis. That's part here, covered in sticky mucus, so they can sort of transfer food into the mouth. So this is kind of a, oops, actually, you know, the sound, I'm just going to stop sharing my screen because I think I didn't turn on the sound for the video, okay? And I'll reshare, because otherwise you won't hear what they're, just any sound on it. Application share screen, okay, share audio. Now I'll share it again. Okay, hopefully you can see that. Yeah, okay. So this is kind of a cool video of a.
How's the ship doing? The ship is underway.
Yep.
Time noise.
Yeah. Acorn worms are very cool.
Sorry, what?
Acorn worm.
There's no doubt where he's being. Hard to tell where he is, though.
Yeah.
Yeah.
Way up there. Wow. You got that capture, Katie?
Yeah, I got it.
I'll go tighter.
Sorry, I was a little fixated. Yeah, I think they're their own phylum.
Wasn't it the sea cucumber?
I don't think so, but yeah.
It just, what a weird ending to an animal. It just like gave up the rest of its body. I'm done right here.
I'm just
Well,
looking at
that's
it
all the
wondering,
filtering that they've been doing, that it's been doing.
do you think they're processing
Yeah.
all the way to the
Been
end
living
there?
here for a while, I'd say.
Impressive, huh?
Looks like he's just lounging on the rock. Like that was all one sitting.
Stop.
It's just like so much dirt in my stomach.
Let's go wide, Alex.
You got it.
Yeah, there you go. It's quite cool. I quite like the commentary on that video. Hopefully you got to hear that. Okay, let's see if I can. There we go. Okay. Okay, so within, I mentioned that within the hemichordates, we had those acorn worms. There's also this group called the pterobranchs. It's a silent P with about 30 species. These are sessile organisms. That means they don't move around. Quite small, okay? Up to 12 millimeters long. So, you know, just a centimeter or so, that live in these tubes that are secreted by the proboscis, okay? Some are solitary, and some, like in this diagram here, are colonial, okay? So they produce lots of them and all join together. And the collar has 1 to 9 pairs of tentacles for prey capture and gas exchange, okay? So that's these parts up here that they use for feeding and gas exchange. And these are actually, when you look at these things, you sort of think, well, you know, when we were doing the protostomes, we were looking at something that looked a bit like that. And there are, you know, some of these worm-like things that look the same. This is a picture of a bryozoan that we looked at when we were doing the protostomes. Okay, and they've got this, you know, the colonial. They've got this feature here with these tentacles out the top. But they are evolutionarily completely distinct groups, okay? These are in the protostomes, these are in the deuterostomes. So this is a good example of so-called convergent evolution, okay? Where groups have gone down different evolutionary paths and then evolved towards a common solution to a problem or a way of life, basically. So independent evolution of similar features in phylogenetically different groups of organisms. Okay, a well-known example of that is also evolution of wings in insects that we talked about, but they evolve completely independently in birds, okay, and in bats, etc. Okay. Okay, so we've talked about, we're still in this group now of the ambulocrarians. We've talked about the hemichordates, these sort of the acorn worms and terebranchs. Now I'm going to talk about the echinoderms, which is a bigger group of living organisms. Okay, that translating from Greek as being spiny skin. And some of their key features are that they have ciliated larvae, as is common to these ambulocrarians. But they're kind of weird, right? Because this is a group, both the deuterostomes and the protostomes are part of these bilaterally symmetrical groups of organisms. But as we mentioned a few lectures ago, these echinoderms actually have radial symmetry. They've kind of flipped the switch back again and reverted to a form of radial symmetry, okay? And they've also sort of ditched the phalangeal slit as well that is common to the rest of the deuterostomes. These are the major groups of echinoderms that live today. There used to be a lot more groups. They were very common in the fossil record, and because they fossilize well, they've got sort of a calcareous skeleton. But these are the major groups that are living today. The sea lilies and sea feathers, a fairly small group, but were very common in the fossil record. They're known as a group called the crinoids. Sea stars, the asteroidea. So somebody asked last lecture or the one before about what's the difference between a sea star and a starfish. Nothing, it's the same thing. Sea stars is supposedly more proper, but it's the same thing. Brittle stars, the Ophiuroidea. Sea urchins, which is the echinoidea, and the sea cucumbers, the holothurioids, okay? So we'll be talking a little bit about those. Now, the body axis in echinoderms is a little bit strange. Because they don't have the bilateral symmetry, instead, people refer to them the oral side, which is the side of the animal that has the mouth on, and the aboral side, which usually is the side that has an anus on, but not in all circumstances. So, for example, this is the schematic of a brittle star that's on the seabed. This is the oral side. Okay, that's the key term to this. This black bit here. And this would be the aboral side. Now, the brittle stars are actually an example where they don't have an anus. The mouth and it is just a single opening to the digestive tract. That's not true with the other ones. So a sea star, again, the same, mouth against the seabed, the oral side versus the aboral. With sea urchins, the mouth is on the bottom, and this whole sort of surface of this like ball of them is most of that is the oral side, and then the aboral when the anus is on the top there. Crinoids, the sea feathers, have an oral side that's actually facing away from the seabed, and the anus is on that same side as well, okay? And then with sea cucumbers, they've done a funny thing where they've sort of tilted on their side and become like worm-like again in a way. So the oral side is actually, you know, at one end of them, rather than against the seabed, and the anus is at the other end, okay? So a strange arrangement to the animals. So some unique features of the echinoderms. This radial symmetry. Are podia modified cilia? No, they're not. I'll talk about the podia in a minute, okay? So the aboral surface is the anus, right? In most situations, that is correct, except in, I think it's just the brittle stars that do not have an anus, okay? And I think the crinoids, I believe, the anus, I think it's in the notes actually, the crinoid, the anus, in sea feathers, the anus located on the oral disc near the mouth, okay? So they're an exception as well. Yeah, so those are the two exceptions. There's notes in the below the slides on that. Radial symmetry, calcified internal plates are features of the group, and the water vascular system that has as part of the podia are part of that, Rodney. Okay, so we'll talk about that. And the type of symmetry they have is called pentaradial symmetry, where penta means 5, and so typically they have, you know, 5 like this, okay? 1, 2, 3, 4, 5, like a wagon wheel sort of thing, symmetry. Now that's not true with their larvae. They retain a bilateral symmetry in the larval stage, this diagram on the left, but they undergo this tremendous metamorphosis as they change into the adult form that takes on that bilaterally pentaradial symmetry. So here's an example of how that works. Here's a sea urchin, okay, a type of an echinoderm. And when you first look at them, so this is a live one that's got spines coming from its called a test. It's like skeleton that they have. But in the dead one, when these spines are off, that's this picture here. You can see there's 1, 2, 3, 4, 5 areas there. And each one of these little tracks that you can see here is actually, those are little holes in the test that the podia stick through along these lines here. Okay, the podia are these little feet that they have. So clear pentaradial symmetry here in the sea stars, the brittle stars, and then sea cucumbers, again, it's not very obvious, but this is in this species, you can see there are these rows along here that have these podia, these feet here, and there's one there, and then there's one there, and if you looked around the whole thing, you would find 5 of these paired rows going along it with the pentaradial symmetry, with the mouth at one end and the anus at the other. I don't know which is which in this thing. Okay. Second unique feature of the echinoderms is this calcified endoskeleton. So it's an endoskeleton, even though it seems to be around the outside of it, because it's covered in a dermis. It has this overlying dermal tissue, thin tissue layer that produces that skeleton. And the skeleton is made of these sections called ossicles that are calcareous. They're made of calcium carbonate, but also with some magnesium carbonate within those. And in different groups, they appear in different forms. So in the sea urchins here, this is various different urchin tests, shells. You know, it's like this ball, basically. In a sea star, if you touch a sea star, they're generally quite sort of hard, you know, sort of spiky, although you can squish them a bit. That's because they have these ossicles within the outside layer of them, okay? Here, this calcified ossicle here. Now, with sea cucumbers, it's less obvious. They're actually, the endoskeleton means inside, yes. It means inside because it's covered by this overlaying dermis, okay? Even though it's like a sort of around most of the organism, right? It still has a layer of tissue over the top of it. Sea cucumbers, it's less obvious. They have these things called spicules, which are really tiny in most cases. But if you sort of dissolve away the rest of the tissue of a sea cucumber and then look under a microscope, you see all these fantastic shaped features that are a little bit, you know, like looking at snowflakes in a way. And some of those are really diagnostic of, they're used for species identification. So echinoderms also have other features as part of that endoskeleton. They have some features called the pedicellaria, and then spines. Now, these aren't in all of the groups, but pedicellaria are like, this is one here, for example, okay? These are pedicellaria between some spines, and these are little pedicellaria, and these can actually act like little pincer-like things, depending upon the species. This is a great one here. This is a crown of thorns starfish. Okay, in the comments below the slide, it tells you what each one of these are. Spines are most obvious in the urchins, of course. This is a purple urchin. They're common on the west coast. Are the spines poisonous? It depends. So on the species, whether they're venomous or not, okay? This is a really spiky species. If you go like snorkeling down in Florida or in the Caribbean or something, the species is called diadema, got these really long spines that are really easy to get pricked on. And this is a pencil urchin where these spines have sort of adapted into much thicker features. But they can also occur in sea stars. So this is a crown of thorns on the Great Barrier Reef with these big spines. And this is Asterias, which is a species, or at least a genus, that occurs on the East Coast here, these spines. Okay, so what is this water vascular system in the podia that we were talking about? Now, this is a really sort of cool system that's unique to the echinoderms, and it's like a bit like the fluids in the brake system of your car or something like that. So it's a hydrostatic forces it's based upon. And just look in this diagram here of a sort of cutaway of a starfish. There's this blue like plumbing, basically. This blue system here that's all linked eventually round to this feature on the aboral surface of this sea star called the madreporite. Now, if you pick up a starfish and you look on the top side of it, the aboral side, often you see like a little sort of calcareous ring that's distinct from the other areas. That's this madreporite, and that water can enter this kind of plumbing system inside. I think it's actually a ciliated feature. And the water is connected by a calcareous tube that goes down from the madreporite. It's called the stone canal. And then it's connected with this ring canal that goes around the stomach and the esophagus of this of the sea star, and then eventually connected out into these radial canals, they're called, going right out to the tip. Okay. Now, all of this plumbing system I'm referring to, that's not its official name, is linked to these podia. That's these little blue features here. Okay. These are little tubes that can expand through hydrostatic pressure. So you can, just like we were talking about when worms move and burrow in sediments, etc., they can squeeze some radial muscles or longitudinal muscles that can force liquid and make these podia extend or retract. And that's also controlled by these ampullae, which are these little systems here. So when these little balloons contract, it forces water out into those podia and they extend. So it's a really cool system, and it's used for food and waste transport, but also for respiration and locomotion, okay, especially. So here's how they're used for locomotion. This is the tip of an arm of a sea star. These are these podia that I was pointing out that are connected to the ampulla. They can extend or retract, and in some groups, such as the sea stars, they have these suckers on the end of them. And so they can be used for like walking along the substrate, okay? This is a cool video. There's a number of videos I've included in here that I'll let you watch yourself in your own time, okay? This is a sea star feeding on mussels, okay? Bivalves, we talked about those in the protostomes. That's one of their favorite food items. And they can wrap themselves around, grab those with their podia and just open up the shells a little bit, and then force their entire stomach inside, release digestive juices, and eat the thing and just leave behind an empty shell, okay? That's a cool video you can check out later. I'll try and move on from that. Okay, so we've talked about some key distinctive features about echinoderms. Now we're going to talk about some different groups of echinoderms. I mentioned these 5 groups earlier on. I'm going to talk about the crinoids first of all, okay? These are the sea feathers. This is a picture of one here, and the sea lilies. They're a fairly small group of living species today, about 600 described living species, but they're very common in the fossil record. This is an example of a fossil here from about 300 to 500 million years ago. And it's thought that these represent a sort of similar to what was probably the ancestral echinoderm feature. The main difference between the sea lilies and the feather stars is that these sea lilies are attached, they're sessile, They're attached into soft sediment in the seabed, and then have these arms raised here. So remember, the oral surface of these crinoids is facing outwards. The sea feathers, however, have some limited mobility. They have these little arms that they can cling to the surface, the substrate with, but they can move around a little bit, and sometimes actually they release and they drift away, and then they'll settle somewhere else. And the way that these things feed is that they basically are suspension feeders on plankton. That means that a little bit like being a filter feeder, basically. So there's small bits of living planktonic organisms in the water column, and as the water moves by, they extend their arms here so that they can use that to capture small amounts of food. And so they have the arms facing in the direction of the flow. So the flow of water is going like this. These are capturing the food, and that's passed into the mouth. So on this diagram, the water flow must be going in that direction. Okay. So the passive suspension feeders, and they have cilia and podia, those tube feet that they use during feeding. The second major group of echinoderms is these asteroidea. So these are the sea stars. Now, the first thing I'll point out here is we talked about how echinoderms have pentaradial symmetry. If you look here, so I can see 5 arms there, 5 arms there, but you know, there's a few weird ones, right? Some groups of sea stars have actually sort of taken it to the nth degree, and they've kind of added on to this basic underlying pentaradial symmetry. So again, another sort of addition beyond what we sort of classically think as just being a single echinoderm feature. About 1,500 living species. If you go down to a rocky shore, you're likely to see these things. Their gonads and digestive system are located, they extend into the arms of these things. In that diagram that I showed with the water vascular system, if you look at that diagram, you'll see that as well. Tube feet for locomotion, feeding, and attachment. So they have suckers on them. If you try and lift a sea star from a rock, you know, they're kind of you have to pull them a little bit to get them off because of those suckers on them. And most of them are predatory. If you go back and watch that video of the sea star feeding on the mussels, you'll see that. Okay. And they're benthic, so that means they live on the seabed as opposed to floating around in the water. They also have this weird ability where a lot of them can regenerate an entire body from parts of their body. Some of them, if you cut off an arm, they can regenerate an arm. A few of them can actually regenerate an entire new individual just from an arm. And this was actually sort of problematic initially when people were, you know, maybe 100 or more years ago, when people were trying to raise clams, etc. And these were a big predator upon them. They go out and they get these things and chop them all up and throw them back in again. But actually, they were just making things worse because those things would then all generate into more sea stars, basically. Sea stars are also, a particular sea star in particular is also a great example of a so-called keystone species, something that we'll talk about later on in the course as well. And the concept of a keystone species came initially, the first time it was coined, came from studies upon sea stars on the west coast of the US. So a keystone species is a species that has a disproportionately large effect on the communities in which they live. And the disproportionate effect of this particular species in the genus Pisasta is that they're major predators of mussels in intertidal rocky habitats. And so what they do is they stop the rocks getting completely overgrown with mussels, and that makes space for a whole bunch of other organisms to be able to live there as well. So diversity increases when you have a healthy population of sea stars. But if you remove those sea stars, what happens is that these mussels just like blanket everything and exclude most other organisms, most other species, and take over. So diversity goes down. So inclusion of Pisasta in that rocky shore community has a big effect upon what the community actually looks like. The third group within the echinoderms are the brittle stars. These are kind of you know, starfish looking like, but they're quite different. They have this disc in the center, and all of their organs are contained within that disc. They don't have an anus on the aboral surface, as we mentioned earlier on. And then they have these long kind of articulated arms. And so all the organs central disc, they're opportunistic scavengers or detritivores. That means feeding on dead organic matter on the seabed or floating around in the water column. And some of them are suspension feeders, where they have, in some areas, so I've been diving in Scotland quite a lot, and you see this a lot in Scotland. You have a huge area of the seabed that's just covered in thousands and thousands of these brittle stars. And they got their arms up in the air like this, and they're catching food that is floating by in the water column. They also have a weird feature, which is, I think, unique to them, or people don't really know, but about 20 years ago, people discovered that, so some of these brittle stars have light-sensitive cells in their epidermis or near their epidermis, so they can see effectively, or at least detect light. Now, when those species that have light-sensitive cells, part of their endoskeleton has adapted in a way so it's shaped like a lens, and it effectively acts like a lens, so it focuses the light upon that light-sensitive area of the organism. So it's acting like a lens, basically, a sort of really strange adaptation of that endoskeleton. And the fourth major group are these echinoidea, the sea urchins. Now that actually also includes what's known as heart urchins and sand dollars. Okay, they're just modifications upon a common theme, basically. So they have this ball-like structure if you're a sea urchin. If you're a heart urchin, they're actually a little bit sort of flattened and elongated. And then sand dollars are like flattened right down, okay? They have spines for defense, and they can use them for locomotion. So some of them have different types of spines that do different features. This purple urchin, a West Coast species, has got these spines for defense. Heart urchins and sand dollars have much smaller spines, and they're softer as well. And okay, so these urchins here, they typically scrape algae that's living on the surface of rocks, and that's their main food source. Whereas these heart urchins, what does it say about them? They live in sediment, deposit feeders. So yes, they're going through the sediment. These are normally buried within the sediment, or almost buried within the sediment. And they're feeding on sediment and organic matter in the sediment. Whereas the sand dollars are actually feeding on planktonic organisms in the water column. Another unique feature about this particular group of echinoderms is that they have this incredible mouth apparatus called the Aristotle's lantern. This is a picture, so an urchin that's been cut in half with its organs taken out. And this is the Aristotle's lantern. It's a really, really complex, it's this cool thing to dissect and look at, actually. It's a really complex arrangement. And this video here, this is only 39 seconds. So let's take a quick look at this. There it goes. This shows you, so here we're looking at the oral surface. So like a urchin on a glass plate here. And describe scraping algae-like things, where they can scrape algae off a rock surface. Very characteristic of sea urchins. So sea urchins can also have a large effect upon their ecosystems. So on the west coast of the US, you get these enormous, if you've ever been to the west coast, somewhere like Monterey or something, and you look out on off the coast, you'll see areas of floating seaweed and kelp. They're incredibly long, and they create these little floating bits on the surface. And that creates a habitat for all sorts of other things, types of rockfish and things like that. But that kelp is fed upon by sea urchins. Now, about 150 years ago or so, people started hunting sea otters on the west coast, and they hunted them almost to extinction for their fur. They have really dense fur. And what that meant was these otters feed upon urchins. You had an explosion of urchin numbers, and in some areas, this kelp was basically, pretty much lost because all of these urchins were feeding upon it. It changes the habitat, and so you get a whole shift in species diversity there. Otters are a keystone species. Now, in recent decades, when otters have been protected, their numbers have rebounded in many areas, and sea urchin numbers have gone down in response. So you get more kelp, and it changes the ecosystem in a dramatic way. So these urchins are very important sort of feeders on kelp and changing the ecosystem. Okay, the fifth group of echinoderms that we're going to talk about are the sea cucumbers, okay, the Holothuriidia. Did you mean urchins are a keystone species, or the otters are? Well, really, they describe otters as being the keystone species, because if you remove that, they're the ones that are removing the urchins, okay, in the way that when we talked about earlier on, the sea stars on the rocky shore were removing the mussels that then changed the system. So people really talk about the otter as being the keystone species, because it's modifying the urchin numbers, which then modifies the rest of the habitat, right? So sea cucumbers. We talked about how they lack arms, but they actually still underlying that have this pentaradial symmetry. It's just they've turned it all on its side and gone, worm-like again. So they have pentaradial symmetry in terms of the way the podia are arranged along their bodies. So they have a mouth at the anterior end, anus at the posterior end. In other words, the oral and aboral surfaces. About 1,400 living species, and most of these live on the seabed, as in the other species we described. But there are some species that are pelagic. That means they live free-floating within the water column. They're all marine. Remember, all of these echinoderms are marine. So they can be suspension or deposit feeders. So suspension feeding is like filter feeding, like this bottom one here that has these. So some of these species live on the seabed with parts of them buried, and then they stick up these tentacles, sticky tentacles, that they get food as it goes by. Others are deposit feeders, like this one here, where they go on the seabed and eat sediment basically and process it and then poop it out the other end. Okay, and they take out the organic matter that's good for them. 5 rows of tube feet along the body, so there is underlying pentaradial symmetry, even though it doesn't look like it. And some of them have a weird kind of a defense as well, with these so-called Cuvierian tubules. So these are like long, sticky tubules. So if you sort of pick up this thing and start messing around with it, it just like blurts these sticky things out. They're toxic as well. Holothurin is the name of the toxin, which is found in the body wall of some of these things. And in some South Sea Islanders, they macerate the bodies of these and use that toxin to catch fish, etc. Okay, so this is a video you can watch in your own time of these sort of releasing of these incredible tubules as a defense. This video you can watch in your own time shows the feeding using the tentacles, where it sort of grabs food from the water column, tiny food parts, and puts it in its mouth. What do you mean? What does macerate mean? Mash up. Macerate means mash up. Okay. Okay. So I'll let you watch those. Okay. So let's see how we still got some time left. I just want to know where we are. Okay, we've got about 15 minutes. And if we get through this, we can look at some of the videos as well. Okay. So let's just take a step back and remind ourselves where we are. Okay. Today we started talking about the deuterostomes, and what we've been talking about so far, an introduction to the general features of the deuterostomes, and then we've gone through these two top groups that are both ambulocrarians. Okay. They have ciliated larvae. The echinoderms have pentaradial symmetry. This group of hemichordates, the acorn worms and pterobranchs have they're like a few species or a small number of species of worm-like marine worms, basically. We're going to go through an introduction to this bottom clade here of the chordates, okay, which is what we'll be focusing on next lecture. And key feature of those is they have a hollow dorsal nerve cord, a post-anal tail, and a notochord. So I'll go through those features. Okay. We'll also talk a little bit about the lancelets and the tunicates, basal groups of chordates. Basal means closer to the ancestral form, and then later we'll go on to talk about the vertebrates. So three key chordate traits, okay? The first that I mentioned was this fact that they have a dorsal hollow nerve cord. What does that mean? If you remember when we were talking about the protostomes, and I showed you a diagram at one point, I can't remember, possibly when we were talking about annelid worms, we talked about how they had a ventral nerve cord, a nerve cord that surrounded the esophagus and then went down the ventral side of the animal. Well, in the chordates, they have a dorsal nerve curves, a nerve cord. So, like our spine or goes down our back, our dorsal side. Okay. And the hollow part comes from that occurs or originates in the embryo. So here, this is just showing how that hollow nerve cord comes about. If you imagine this as being the surface of an embryo, you get this along, imagine down our spine as we're in an embryo, you get some tissue that folds in, okay, and creates this little pocket area here, okay, which eventually just carries on folding in so that these two bits of ectoderm come together and fuse, and now you're left inside, down your back, basically, this hollow nerve cord, okay, the neural tube. So developmentally, it's different from, for example, that ventral nerve cord that occurs in, annelid worms, for example. So this lineage, prior to chordates, have a ventral solid nerve cord. And then it becomes the brain and central nervous system. So, going down our spine, that would eventually, the anterior end would develop into our brain as well. The second unique feature of the chordates is the notochord. So what is the notochord? It's not the spine, it's not the same as the spine, okay? It's not a bony feature. It goes down the dorsal side of the animal. So I'm just going to flick back to that picture I was showing you before. Okay, well, we've got the same picture anyway. Okay, so here's the hollow dorsal nerve cord. It underlies that. Okay, this notochord here. And it's a core of large turgid cells with fluid-filled vacuole. So what does that mean? Turgid means that it has fluid inside of it and that it's kind of under pressure, okay? That makes it stiff. So effectively, this notochord is acting like a stiff rod down the dorsal side of this animal here, and that allows it to get mobility because there are muscles attached down the side of that. And if you imagine a stiffened rod and you have muscles that pull on one side of it, what it does is it causes that causes it to bend one way as opposed to just collapse floppy. Okay, it causes it to bend because it has some rigidity. And if you relax muscles on one side and then contract on the other, it'll go the other way. So it's allowing it to get this kind of motion that can be used for swimming. It's non-compressible because it's fluid, you know, has fluid, non-compressible, so it bends when muscles contract it. In vertebrates, a derived form of chordates, it is ultimately replaced by the vertebral column during early development, okay? But nevertheless, it typically appears in early embryonic or at some stage during development. So retained by some chordates, such as the lancelets and tunicates that we'll talk about, and some primitive types of fish as well. Important in embryo stage, coordinate signaling. Okay, so it also has some other functions in terms of embryonic development. There are signaling chemicals that are released at different points along it that control development on an anterior to posterior axis. We talked about there being three key traits of the chordates. The third one is the post-anal tail. So what does that mean? So here in this schematic, you can see here's the digestive system, and the anus comes out here. All of this region here is a tail, okay, that is post-anal. In other words, it's occurring in the posterior end of the anus, okay? So it's an extension of the body past the anal opening. In all chordates, sometimes only embryonic stage, such as, I don't think I've got a tail the last time I checked, but, you know, when I was an embryo, I would have done at some stage, okay? And it's an adaptation for rapid swimming. So it's sort of part of, it goes with that notochord that's a swimming adaptation. Chordates also have pharyngeal slits. Now, this isn't unique to the chordates, because remember we talked about they're, for example, in the acorn worms, okay? But they're typically more pronounced. So in pharyngeal slits occur in all chordates. So remember, this is our pharynx, like the throat area, and the slits are openings that allow water, if it comes through the mouth, it can come out through the slits back outside. Often lost, highly modified in adult stages. So I do not have gill slits because of that reason. Also occurs in hemichordates, as I mentioned, but it's been lost in the modern day echinoderms, so they do not express them. Originally, they occurred as an adaptation for feeding, filter feeding, but there's been many modifications in the chordate since then. So after they originated in more derived forms, there were gill arches that formed. This is supporting bits of bone, basically, that support the gill arches. And there's been many modifications in those in more derived forms that lead to things such as our jaws. Our jaw bones are, and our ear bones are modified pharyngeal slit gill arch supports, okay? We'll be talking about that next lecture. So, two of these basal groups of chordates, basal meaning less derived, closer to the common ancestor, okay, the lancelets and the tunicates. Lancelets is a small group, only 35 living species, and they occur in marine, warm coastal areas and some areas. And these They look a little bit eel-like maybe, but not really. But they actually have no obvious head or eyes or anything like that. They're buried in the seabed with just the anterior end sticking out. And what they do is they take in water and then pass it through the pharyngeal gill slits partly for gas exchange, but they're filter feeders as well. So these are quite small things. What does it say? Less than 5 centimeters, so this big or smaller. And so they're just filter feeding, and they have a notochord. The adult forms of those have a notochord that is passing around down, remember, the dorsal side of the organism, just underneath the dorsal hollow nerve cord. The second group, so this one here, the tunicates, also called the urochordates. These include things like sea squirts. So if you go down to the seabed, down to the ocean, you pull up a rope, for example, that have been lying there. Often you see these sea squirts attached to like a rope or something. Of these jelly things, those are sea squirts. There are other types as well called salps, a colonial planktonic but quite large organism, and larvaceans. But these sea squirts are what we would most typically sort of see if you went down to the ocean. These are marine filter feeding organisms that look nothing, this is what's bizarre about them, they look nothing fish-like or anything like that. You wouldn't suspect that they're chordates and, you know, in the same as we are chordates. So the adults show no obvious chordate characters, except they do have pharyngeal gill slits that they use for filter feeding, but they have no notochord. But the relationship becomes more obvious if you look at the larval stages of these tunicates, okay? Let's just compare here. So this is a sea squirt, an adult stage of a sea squirt. And they have this siphon here where water, they can pump it in to this chamber here, and then it goes, they pump it back out of this X-current siphon, it's called in this diagram. And as it does so, it goes through some pharyngeal gill slits. Okay, that's how it filter feeds. When you look at the larvae of these, this is a mobile stage, the larva. This is sessile, it's attached to the seabed. They have a notochord with a hollow dorsal nerve cord. They have a post-anal tail, and they have pharyngeal gill slits. And these things can sort of swim around a little bit like tadpoles, basically. And eventually, when they settle and metamorphose into these adult forms, this anterior end will attach to the seabed, and then they go through this sort of this wonderful transformation. So it's this larval stage that, when you look at it, you really see, you know, the common chordate features within those. Okay. So, okay, so as I normally do, I put in some summary slides that you can use for exam revision purposes if you're just trying to get some sort of common structure to all of this. Okay, so I'll leave you with those. And 29, so we've got 1 minute for question, or I can show you a quick video, whichever people prefer.
I just had a question.
Sure.
The notochord and the nerve cord are different things. They're not the same.
They are different things, yes, entirely different things. So let's just take a look at this diagram here. So the nerve cord is, you see these cells here, okay? That's the nerve cord. That's cellular. These are cells that become, nerve cells, basically. And that's formed by this endoderm that like folds in like this and eventually becomes distinct and separate like that. The notochord is different. That's this here. So this feature is distinct from that, and it derives from different embryonic tissues altogether. Okay, I can't remember. I think it's mesodermal origin, but I don't remember exactly. Whereas this has got a, an ectodermal origin. Okay. And then these are like long cells that are like really rigid and stiff. Okay, this notochord. So the notochord is ventral to the immediately ventral to the dorsal hollow nerve cord. Does that make sense?
Okay, so I could just say the notochord is a solid sort of tube that's
Stiff,
ventral
it's like
to
a stiff
the
rod.
nerve cord, or stiff rod that's ventral to the nerve cord, which is hollow.
Correct.
Okay.
Yeah, they're both on the dorsal side, but you know the notochord lies slightly ventral to the nerve cord.
Okay. Thank you, Professor.
Okay. Yep. Okay.
Oh, hey, Professor, I had a quick question.
Sure.
In the echinoderms, the calcified internal plate, I was a little confused. So it's part of the endoderm, but it forms on the outside of the organism?
So it basically has an overlying thin layer of tissue. That's the key feature. It's not like if you were to get a, let's see if I can have a cross section of it anywhere. I don't think. No, I don't. It's but basically, you know, this. Okay. If you're still looking at the screen at all, this test here, this calcareous plates that come from this urchin, right? So this is dead, and all the spines have come out. It's got no living tissue in it, and the calcareous plates are just exposed to the air here, or the water if it was underwater. In this instance here, in the living organism, it's got a living layer of a thin layer of tissue that overlies on top of this kind of bony plate, right? So what is exposed to the water is not the shell directly, there's living tissue on top of it. Does that make sense?
Oh, okay, yeah. That makes sense.
So the dermis is like a thin skin, basically, over the top of that.
All right, thank you.
Okay.
Professor,
Any other questions while I've got people?
Yeah, I had a question about the notochord and the neural tube. Like,
Sure.
what are we supposed to like? I remember we learned about it in 105, but like, what should we know primarily for like their function or how they form?
You know, check out the chapter, but I said pretty much I think what's in the chapter. I mean, the key feature is that this is a. Let me just try and find the relevant slides. But these are features that are common to the chordates, right? That's one of the important parts to it. I'm just trying to get my control panel here so I can find out where these things are. Okay. Yeah, so, you know, some of the key parts in terms of understanding this chapter are to know that this dorsal hollow nerve cord is typical of this group, right? It's not occurring in other groups of organisms. And the notochord as well is typical of this group. It's not occurring in other organisms. So these are synapomorphies of the clade of chordates, meaning it's a feature of the common ancestor that is then perpetuated in or modified in the descendants of that group. Okay. In terms of what they're doing, you need to know essentially what I've got in the slides here, right? That this hollow dorsal nerve cord is on the dorsal side of the organism. It's formed by invagination in the embryonic stage from this ectoderm, and that the notochord is a stiffened rod that lies immediately below that. That's important for locomotion. It's rigid, and also has developmental importance because there are signal chemicals that are sort of associated with that notochord. So those are the key parts that you need to know, okay?
Okay, thank you.
Good. Anyone else got questions while I'm here, or I'm available in 24 minutes for office hours, either way. There's a link for office hours in on Blackboard, okay? Now we'll stop.